Hello, everyone. My name is Zach Strickland, and I'm a senior neuroscience and biology major with minors in chemistry and religion. And my project this semester is comparison of multiple sclerosis models in Danio rerio. And throughout this project, I've been mentored by Dr. Chris Strand and Harshana De Silva Theology. So diving into the introduction, my project is focusing on evaluating literature and comparing different models in zebrafish to allow us to later explore how demyelination and remyelination occur in zebrafish and ultimately investigate what environmental stimuli affect these processes using molecular and behavioral tools. Broadly speaking, these questions have relevance to multiple sclerosis research. For those who don't know, multiple sclerosis is a debilitating progressive demyelinating disease with limited treatment options in humans. So it's very much worthy of further research, and there's a lot of cool stuff happening in that field right now. So to get at these questions, we're going to be using zebrafish, which, while they're not mammals and they're obviously not humans, they're an excellent model system. They have very high genetic similarity to humans. They develop transparently ex utero, which will be very important later on. They have lower costs than murine and rat models. They're very easy to genetically manipulate, which is also super important. And they have large clutch sizes, all of which are very advantageous to us when we're looking at model systems. For this project, I evaluated different model systems in literature based on how they've been previously used in publications, what types of questions we can ask using them, what they will cost us, both in terms of upfront costs and long-term costs, and how easy they would be to implement and maintain at Concordia. So diving in, the first thing we'll need to explore is how we would create a transgenic line of zebrafish. So we'll go through the theory behind that and then go into an example of a transgenic model that I think we could use. So first, to create the transgenic lines, we're going to need zebrafish. So we'll breed them and collect fertilized eggs. Once they're at the 1 to 4 cell stage, we'll actually take them individually, and holding them with a pipette, and looking at them under a microscope, we can actually move a microscopic needle into the egg and inject a transgenic construct. And because they're so early in development, a lot of these constructs will be taken up into the DNA and can actually be expressed then. So once we've done a bunch of these injections, we'll screen out embryos that are not expressing the transgene. Our transgenic construct will be labeled with a fluorescent probe that we can see visually. So we'll basically look for the colored eggs, and if the eggs are colored in the way we're expecting them, we'll keep them, and if they're not, we'll discard them. And then we'll raise the colored eggs up to adults and then breed a stable line. And then we can do experiments on those fish or those larvae. So for a transgenic model system for remyelination, demyelination research, we will look at the metronidazole nitroreductase system. Metronidazole is a drug that's non-toxic to zebrafish unless the zebrafish is expressing the bacterial nitroreductase enzyme. So by strategically placing the nitroreductase enzyme under control of the myelin-based protein promoter, we can express nitroreductase only in the cells that myelinate, which allows us to specifically target oligodendrocytes, which is super powerful because we can then dose the tank that we're treating the zebrafish in with metronidazole and just put the fish in the tank and watch them, feed them, do water changes like we would normally. But all that time they're being exposed to the drug, and then only the cells that express the nitroreductase gene will convert that non-toxic drug into a toxic product, which will result in ablation of those cells and in demyelination. So this works because nitroreductase will convert the prodrug metronidazole into a cytotoxic product and basically remove any cell containing the enzyme, in this case, oligodendrocytes, which will result in demyelination. So once this has happened, we'll put them in the tank for about 4 days at 5 millimolar concentration. We'll transfer them into a recovery tank and monitor their recovery based on behavior. And we can also monitor their recovery by sacrificing fish at desired time points to visualize how the myelin's responding across time using confocal microscopy or immunohistochemistry. And this can be a really powerful system because, again, we're specifically able to target the myelinating cells by placing the nitroreductase gene under control of the myelin-based protein promoter. If we weren't going to use a transgenic system, we could do a microinjection of a toxin model, of a toxin. In this instance, we could use LPC or EAE. I will get into what those are after we talk through the microinjection process. So to do a microinjection, we're going to anesthetize the fish so it's not going to feel any pain during the surgery. And then we're going to do a small surgery to expose the spinal cord, and then we can move in a microinjector needle and inject a very small amount of the toxin into the spinal cord to allow us to watch how demyelination happens. The fish will be knocked out, it won't feel any pain during this. It'll be secured with these sponges like we see in the graphic here, and then the microinjector will put in our toxin. Then we'll move it to a recovery tank and watch how the fish responds behaviorally, and also using immunohistochemistry, Western blotting, or fluorescence microscopy, which would require sacrificing the fish, but would give us more direct knowledge of how the myelin's changing. So I mentioned LPC or EAE. So what are those? The EAE model stands for experimental autoimmune encephalomyelitis, and it's another commonly used system for looking at MS-related questions. And this works by injecting myelin oligodendrocyte glycoprotein, or a protein into the zebrafish that sort of targets myelin and reacts with myelin, which causes the immune system to respond to the new protein. This causes the blood-brain barrier to become permeable to leukocytes and blood proteins, which results in T cells and B cells going into the, crossing the blood-brain barrier, going into the fish, interacting with each other, and then causing this inflammatory cascade by releasing chemokines and cytokines, which results in increasing inflammation around the neurons and the myelin ultimately being removed. Additionally, we could use LPC, which is lysophosphatidylethylamine. It's a mouthful. This is actually a toxic detergent. Injection of this into the subdural space causes focal demyelination, so demyelination in one specific spot, because LPC actually causes the myelin to sort of shrivel up into spherical vesicles, which are then taken away by glial cells, resulting in literally the removal of myelin, so demyelination that direction. And both of these result in demyelination and allow us to monitor demyelination and remyelination. One benefit of the LPC model is that it doesn't rely on immune system involvement to do the demyelination, so we have very precise spatial control. So exactly where we put the LPC is exactly where the demyelination is going to occur. With the EAE model, it relies on the immune system involvement, so it's a little bit less precise for the spatial control, but both still allow us to look at how the remyelination would occur. All right, so in conclusion, that's a pretty broad review of how we would create transgenic lines and how we could microinject different things into adult fish. So in conclusion, each model presented here offers unique challenges and benefits to our research group. However, the metronidazole nitroductase system will minimize long-term costs while allowing for comparable data collection. The flexibility and ease of using a treatment tank for each trial instead of a microinjector for each fish is very beneficial because it will allow us to treat multiple fish at one time versus having to remove and anesthetize each fish and do a small surgery on each one. So it's going to be a lot less labor intensive when we get to the point where we're actually doing these trials. And while it does require a microinjecting system to create the transgenic line, we could outsource that to a core facility somewhere, like the University of Minnesota or the Children's Hospital of Philadelphia. Both have zebrafish core facilities capable of doing this for outside research groups. And outsourcing that's actually a pretty relatively simple process once we have that transgenic construct created for injection. So our next steps ideally will focus on designing and synthesizing this construct that we would send off to be injected into wild type embryos or eggs. And additional work could also focus on developing an in vitro model of demyelination using cell culture while we work on getting the in vivo zebrafish model set up. So in closing, I would like to thank the Office of Undergraduate Research, Scholarship, and Creative Activity for funding this internship, and Dr. Strand and Harshana Salafilaji for mentorship and advice throughout the project. Thank you so much for watching this presentation, and I can't wait to answer any questions you have during the live Q&A period on Wednesday. I'll see you then.